Greetings and welcome to this month's Tipping Points webinar. I'm Robert Inglis and I represent Jive Media Africa. I'm one of the directors at the Specialist Science Communication and Research Engagement Company. And we are truly excited to be working with Oppenheimer Generations Research and Conservation to increase the visibility and impact of research going on the continent. So welcome to Tipping Points. It's episode 34. We're asking key questions about how to preserve biodiversity while balancing conservation and development. This webinar series is an initiative of Oppenheimer Generations Research and Conservation, and it takes place on the last Thursday of every month at 1 p.m. So please do keep tuned. I'm so excited to be here with all of you today because today we are going to be meeting the top 3 candidates from the Jennifer Ward Oppenheimer Research Grant. This is a very generous grant of $150,000 US awarded to one researcher each year who aims through their work to cause positive impacts on African landscapes. There are some fascinating topics on the table, and we're going to meet the people who are driving these ideas forward. But before we meet the panelists, I just want to make sure that everyone's aware that this session is being recorded. It will be available on the OGRC Oppenheimer Generations Research and Conservation YouTube channel. And this is also where you can catch up on other webinars that you may have missed. And I also encourage you to sign up for the OGRC quarterly newsletter and stay tuned to OGRC's social media channels so you don't miss upcoming webinars and announcements and topics related to conservation and development in Africa. If you're interested in the JWO research grant specifically, please also sign up for the Jennifer Ward Oppenheimer Research Grant newsletter. We're excited to have such a great turnout today. Please continue to introduce yourselves in the chat box and engage in the chat box. And with a slight variation from previous editions, this time there's also a Q&A function. So if you'd like to put a question to one of the panelists, we'd love to hear what you want to know, and put questions in the Q&A function, chat to each other in the chat, and let's have an engaging and entertaining space that we're here today. So I'm really pleased to introduce you to the JWO top 3. When applicants were called for the JWO grant, there were 848 applicants from 41 countries. And this is just a testimony to how this initiative has grown over the last 7 years. There were 7 candidates who were named as finalists, and you can read their stories on the JWO website. And today we're going to meet the 3 top candidates, and you're going to have a chance to hear what they're proposing to do if they are to win the research grant. And also you'll be able to ask them questions to learn a little bit more about them. So without further ado, the 3 top JWO contenders this year are Beatrice Nganso, Levi Otwamo, and Nompumelelo Basumdiza. And we're going to hear a little bit about each of these panelists before they speak. All right, so first of all, we have Dr. Beatrice Nganso. Beatrice is a research scientist under the Biodiversity and Environment Thematic Research Program in Kenya. Her research examines the health and diversity of insect pollinators as they interact with plants and biotic and abiotic stresses to improve ecosystem resilience, while empowering stakeholders, including young people, as active conservation partners. So Beatrice, over to you. We'd really love to hear what you've proposed for the Jennifer Ward Oppenheimer Research Grant application.
Thank you, Robert, for the nice introduction, and greeting to everyone joining this webinar. So let me share my screen. Okay, greetings once again. My name is Beatrice, and my JWO proposal focuses on building African first open access plant pollinator network database to assess network resilience, but at the same time strengthen biodiversity conservation, food security, and livelihood for millions in Africa. So I'll begin by saying that plant insect pollinator network matters because they are the engine of Africa's food production system, ecosystem, and economics and biodiversity. Through their mutualistic interaction, as you can see here, they put food on our table from agriculture that support about 650 million rural farmers. They also strengthen the diversity of wild plants, thereby providing food for wild and domestic animals, but also enhance air and soil quality. At the same time, they diversify livelihood incomes through beekeeping. But then insect pollinators are under pressure from these environmental factors, but we know very little about their identity, their distribution, and even the forage plants that support their health and productivity. At the same time, despite Africa's vast land area, as you can see here, the continent contribute very little to the documentation of plant and insect diversity in this famous open access database. And this open access database are also limited in the sense that they do not report species interaction network. Also, an open access continental dashboard I recently developed that showed the distribution of bee forage plant across the globe that support the data gap in forage plant documentation in Africa, as you can see, compared to other continents. But at the same time, this map reveal an uneven lack in the documentation of biodiversity, in the sense that you see that the report is mainly championed by South Africa compared to other African regions in recent times. So what I would say at this point is that we are flying blind in Africa, because without robust data on plant pollinator network, we won't be able to design concrete policy at the continental level that support our biodiversity, food security, and livelihood. So what I'm proposing to do is to build African first open database that will integrate this plant insect pollinator interaction data, land use and climate map, but also models that will help us to identify drivers of network resilience for biodiversity conservation, food security, and livelihood. So my approach to this is first to capture existing published and unpublished data through 11 institutional collaboration across all these countries highlighted in red, but also generate new data on plant insect pollinator interaction, but also profiling the forage plant that support insect pollinators in underrepresented region across this country marked in green. So once all this data is integrated, then we estimate resilience for plant and insect pollinators using mathematical model, but also using additive modeling to understand how these individual environmental factors influence this resilience, but also influence one another. But then also going a step forward to use joint species distribution modeling to predict species co-occurrence pattern in study areas that would not occur in our database. So, and now once this data and all the modeling has been done, then we would develop the graphical user interface for the platform. And this graphical user interface would consist of a species page modules that will simply showcase the digitized images of all the plant and insect pollinators present in our established network. We will also have a species interaction modules that will display the interaction networks using standardized vocabularies. We also will have a plant nutritional modules that will simply present the rank nutritional profile of forage plant species along a GIS layer that show their distribution across Africa. Then we have a repository module that will simply integrate all the data that we have sitting in the platform. then the land use and climate map, but also network metrics and models that will compute various metrics and environmental factors that influence the network resilience. So then once the platform is established, then we will disseminate the utility and the result of this platform at both national level and continental level among students, entrepreneurs, national partners, even traditional leaders and EU member state countries. Okay, now my team is highly interdisciplinary in the sense that we have national partners with expertise in entomology. We have international partners with expertise in mathematical modeling, computer programming and simulation modeling, and the regional partners which will contribute the data in the platform with expertise in biodiversity. So the impact of what I'm proposing is that I'm planning to develop a platform that will host over 300,000 plant insect pollinator interaction network to enhance our understanding of biodiversity dynamic in the long term, but at the same time provide the key forage plan that support insect pollinators across Africa so that we can leverage on that to inform climate smart and nutrient-rich pollinator-friendly, inform the development of climate-smart and nutrient-rich pollinator-friendly landscape in Africa, but also building the capacity of postgraduate students and all the institutional focal points that will contribute the data in biodiversity and data science research, and come up with key network resilient indicators that we can leverage on to monitor this pollination system under environmental change at the continental level. And lastly, to develop a manual on biotic interaction data sharing standard that the general public can leverage on to document plant insect interaction data. So thank you very much for your kind understanding.
Thank you, Beatrice. That's really sounds like a powerful platform. I just want to try and understand things clearly. So you're saying that there's a lack of knowledge about these interactions. So we're not speaking particularly about the plants or about the particular insects, but we're talking about the interactions between those that you'll be putting up into this database. Yeah. I guess my question is, I saw you've got a number of partners involved, but who populates this database? How does it get filled up with all of this wonderful information? And then once that information is in place, who uses that information and how do they make use of that information?
Okay, thank you so much, Robert, for your question. And the national partners or all the institutions that I will establish a collaboration with are those that will contribute into these data sets. And they are found, I focus on 11 institutions that I know across Africa have done work in documenting insect plant pollinator interaction. So they are the ones that will contribute will contribute with existing data set. But then at the continental level, we have regions where we don't know much about this insect plant pollinator interaction. And these are mainly those located in West Africa. So Burkina Faso, Ivory Coast, and even Kenya will contribute with new data set. And the interest in Kenya is because there's a lot of invasive plant species which are invading protected areas. So I will also want to see how do they affect the local network. That is my interest in Kenya. So I didn't get the last part of your question, please.
It was who was going to then use this data and how do they make use of this data?
Now, the data will benefit various stakeholders. As we have, I would say the scientists can leverage on this data to be able to understand how various environmental factors affect the plant interaction, affect the interaction network that will be established. They can use it, they can do it even in the long term because it's a platform that will be publicly available for all. And then policymakers can leverage on this robust data set to develop robust policies at continental level that will enhance our food production system, because as we speak, there is a lot of pressure to enhance the food demand by 2050, given that the population, the global population will increase and half of them will reside in Africa. So they can leverage on this because this platform, insect pollinators are key when it comes to production of our food. They pollinate over 70% of the major food crops that support our food system. But also EU member states can leverage on this to tap into new opportunities of carbon credit market, which these are new opportunities when we talk about biodiversity conservation. And for the youth, it will be a platform to learn how to document this to the youth and even the national partners or those that I will work with. This is an opportunity for everyone to learn how to document this platform, because to document interaction data, because as we speak, the available database do not report on species interaction. Meanwhile, species they don't live alone. Okay, they don't live in isolation. They interact with each other. So I'm going through this effort to add African voice in using extension of Darwinian core standard to document this. Yeah, thank you.
Fantastic. Beatrice, thanks very much. And we've got an overview. There is a Q&A, so you're welcome to drop any questions directly for Beatrice into the Q&A, and Beatrice can respond to some of those there immediately. And then also we can take some of those questions in a session after the other speakers. Thank you very much for that presentation. So our next presenter, our next JWO finalist is Dr. Levi Otuma. She is a senior scientist at the Kenya Marine and Fisheries Research Institute. He specializes in marine ecology with a particular focus on combining molecular tools with field-based studies to address species origins in the marine environment. So we're going to hear a lot about eDNA, and he is currently exploring the utility of eDNA as a biogeographic tool to evaluate the human impacts on marine ecosystems. So welcome and congratulations, Dr. Otuma. Please tell us a little bit more about what you've proposed for your research application for the Jennifer Ward Oppenheimer Research Grant.
Yes, thank you very much for that wonderful introduction. Yes, my name is Levi Otuma. I'm a senior research scientist at the Kenya Marine and Fisheries Research Institute in Kenya, located at the coastal city of Kenya called Mombasa. So in my Jennifer Ward Oppenheimer grant application, I've proposed a solution to Africa, some pending problem that has been affecting African countries. And it's basically, I'm basically trying to empower African nation to adopt conservation initiative that are protecting and creating awareness to transparent engagement in Africa. So my project is called Emancipate. And from the word emancipate, it means setting somebody free. Just to give you a glimpse about marine environment or marine ecosystem, globally, fish provide nutrition to a great number of people. In Africa, this is estimated to be around 20% of coastal population that make up to 200 million people in Africa. But there's a significant portion of fish diversity that is currently declining globally and including Africa. And this is because of several factors, which include habitat destruction, overexploitation, and climate change. So this, because of this, because of the significant uh, uh, reason fish does in terms of food production for African nation, there's need for accurate and timely assessment of species diversity and population dynamics to inform evidence-based conservation for management on the African continent. And these efforts need to be synchronized because, as you realized, fish resources and fish species are transboundary in nature. They don't occur in one nation or within the jurisdiction of one country. They go across borders. So we need solutions that are transboundary. That's why in my project I've proposed solution that will be adopted by more than 2 countries. So currently, as I said, that we need accurate methods to monitor fish. The methods that are currently being used, especially on African continent, to monitor fish have a problem, and the problem this traditional method have is include low sampling efficiency, because in the marine environment, the method that is mainly used to assess fish biodiversity, which is underwater visual sensors, tend to underestimate the critical cryptobenthic fish. These are the fish that fuel biomass production in the marine environment. For example, if you see in the picture that is in red here, in that picture there are more than 2 cryptobenthic species, but you can only see one. So if you are swimming in the marine environment and recording fish population and species, with naked eyes we'll only see one species, but in that picture there are more than one. So that's a challenge in terms of fish monitoring. This traditional method are also face a challenge in terms because of habitat destruction, which include the beach seining and trawling, which because this equipment are troll alongside the sea bed and destroy the ecosystem that the fish lives in. There is also challenge of harm to organism because some of these methods involve catching the organism and actually killing it, which becomes even counterproductive in terms of biodiversity conservation of fish. These methods also rely heavily on taxonomic expertise, which is very rare on the African continent. So we basically need to provide a solution or an alternative to this traditional fish monitoring methods because of these problems that I've just mentioned. And eDNA provides a solution to this because with eDNA, this method is very sensitive and very sensitive, non-invasive, and provides a very powerful alternative in terms of detecting the tiny cryptobenthic fish species that I mentioned that powers production in the marine environment. eDNA, despite its solution on the African continent, we find that it has been rarely been used. If you see in the graph, in a study that was done in 2023, in Africa, there is a very tiny portion of studies that have been used, that have used eDNA to monitor fish biodiversity. And this is mainly because the African continent lack well-established lab. You find these well-established labs are located mostly in the global north, and with which are well-funded. Another problem that limits our application of eDNA is because we rely so much on external sequencing. I was mentioning this to somebody that in Kenya alone, if you want to send samples for sequencing, you will need an ABS permit that takes approximately 1 year to do this. You can imagine if this is a master's a project or PhD project, you might not be able to do it if you follow the due diligence of shipping your samples for sequencing. So they also, in terms of using eDNA, we also don't have comprehensive database, because once you do eDNA, you need to scan it across database that is specific to the region. In Africa, we mostly, with eDNA research, we rely on database that has been developed or species that have been barcoded from different regions, which actually reduces the accuracy of our species detection or eDNA research. So my project, what is basically the solution that I'm providing is to try to empower African nations to monitor the marine fish biodiversity on their own. And this, I hope that it will strengthen that, it will make strengthen data-driven sustainable management on the African continent. So this objective will be implemented through 4 work packages. These 4 work packages are interlinked, and they are transdisciplinary in nature. They cut across from social science all the way to ecology and biology, as well as capacity building in nature. So what I'm proposing is actually to try to first, because Africa, we also don't have a problem of linking, science is not correctly linked to policy, or policy makers really do not use our data, our research to develop policy. So in the first work package, what I will do is try to make a forum through hosting workshop do questionnaire studies, focus group discussion, to stakeholders that will be identified through top-down approach and bottom-down approach. So these stakeholders will involve policymakers that are highly top in the sector of fish monitoring, to the lower person that is the fisherman that actually relies on the resources. And here we will try to just understand what are the gaps in terms of policy implementation, policy making, and try to foster a dialogue between these different stakeholders to try to make sure that fish biodiversity protection, the data that is produced, is actually the policy that are made are science-based policy. That will be the first work package. The second package is about capacity building, and here what we will do is train early career researchers and establish networks of these early career networks, and link them to 5 sequencing centers in 5 African countries, in South Africa, in Mozambique, Kenya, and Senegal and Ghana. So in even this training, it won't be a a top-down approach training, but we'll try to do something known as a training need assessment. We'll roll out an online questionnaire across Africa, find out what is the capacity need that is needed on the African continent, then design this training with these inputs, and then roll out the module. And then after the training, these early career scientists will be linked to these sequencing centers, so that it's just not a training and you get a certificate, but then you continue working with experienced scientists in these sequencing centers, so that there's a sustained generation of results that is linked to policy, not just scientifically enriching the forum. Then we will also make sure that on the third work packet, to promote eDNA research in the continent, we'll produce a comprehensive reference database based on mitochondria genome. And this we will do by collecting fish specimen and then keeping voucher specimen in in the 5 African countries that I've mentioned, keeping the voucher specimen in the National Museum of these countries that I've mentioned. And we are hoping that to capture the entire spectrum of marine fish species, and hoping that this database will help discoverable of so many species that have not been discovered on the African continent, and promote eDNA research. Then fourthly, we will try to standardize eDNA methods or protocols that are used on the continent. And this will be based on the portable Oxford nanopore sequencer. As I mentioned, that we have this problem of sending sequencing to different countries, but with the device that we'll use to do both the reference database and standardized protocol for eDNA monitoring, this device is will be as you can see in the picture, you can actually use it in basic condition in the field. So the protocols that we'll develop will be based on this Oxford nanopore, so that the countries where these sequencing centers will be there, they'll be continuous, because this is very affordable to use. To buy one device plus a PC is not more than 5,000 USD. And if there's a combination of projects, it's very easy to run it. And if you have a larger equipment, so this will be what we will do in terms of work packages. And what I hope my project will do is to produce a standardized eDNA sequencing protocol on the African continent, which I believe will be feeding into the international information on eDNA, because Africa, we face a problem of maybe producing data that is viewed as not being standardized. But with the protocols, that we'll have standardized, that are aligned to the international protocol of monitoring eDNA, I believe that the data that we'll be producing will be linked to the international repositories. And then we'll also produce the mitochondria genome reference database, which I think will promote so many eDNA research on the African continent, because the mitochondrial genome reference database will not only be for one gene, but for the several genes that occur in the mitochondrial genome. So we will promote eDNA research beyond just using 12S, but whichever gene researcher will have selected. So this will be a very comprehensive database that will promote eDNA research. Then there will be that network of early career scientists that will be linked to the sequencing centers on the African continent, that I believe will be very key to improve eDNA research on the African continent, because of their link to the sequencing centers that will be developed or will be purchased under the project. As I mentioned, they're using the Oxford nanopore technology. Yeah, and then there will also be publication at the end of the project and the modules, and also a course program for eDNA conservation on the continent. Thank you very much for listening.
Fantastic, Dr. Levy, thank you. And it looks like you've really thought about this thing from a holistic perspective. So can you tell me just one anecdote from your early work in this field that made you feel like this was really the direction that you had to move if we were going to secure fish stocks across the continent?
Yes, if I give you from the perception of it, lacking a well-established sequencing strategy for myself in Kenya, in I believe it was during my PhD when I was doing my molecular work. We collected samples and needed to sequence them in the lab in Kemfrey, but we had a very lengthy period for applying for an ABS permit to ship these samples. I believe it took more than 1 year. Actually, the 1 year that I mentioned was for my project to uh, um, uh, establish the peak. For the ABS permit, you need to establish a peak, MAT, and MTA. This is material transfer, mutual agreement terms, prior informed concept. For just to get the ABS permit, that is the access benefit sharing of the genetic material that comes from there. And there were so many collaborators that were not willing to undertake this. So I thought maybe with these challenges, I thought maybe developing a sequencer for myself in the lab in Kenya will promote so much research, because we have a lot of resources. We have a lot of what is not known in terms of fish biodiversity on the African continent. We have a lot in terms of data. We are, I think, the continent that has a management method that is not well guided by science. So we need this comprehensive information in terms of what is the status of the marine environment, because as I mentioned, eDNA provides a very comprehensive information in terms of what is the status of what is there, other than if you are using methods that are based on naked eye, that miss a lot of critical organisms.
So those technologies are promising huge opportunities and huge possibilities, but they come with a whole lot of infrastructure and capacity building that is needed as well. So I can see you've thought about. Thanks very much. And we will again ask people to drop any comments into the chat for you or into the Q&A section. So please, just for the audience to note, there's a chat, there's also a Q&A, and if you drop things in the Q&A, then the speakers can respond to you directly. Thank you, Dr. Tuma. So our third panelist, finally, all the way to South Africa, is Dr. Nompumelelo Baso, who is a postdoc fellow at the South African Institute for Aquatic Biodiversity. Her work focuses on the interactive effects of invasive species and climate change in freshwater ecosystems. She's also passionate about using data science to tell stories, to inspire solutions, and to support communities across Africa. So, Dr. Nompumelelo Baso, please tell us a little bit about what you've put forward as your proposal for the JWO grant.
Thank you so much, Robert, and hello to everyone. I'm honored to be here. So I'm going to try and give a very quick snapshot of what I've proposed. I've only been given 7 minutes, so I'm going to try my best to stick to the time. So my proposal is looking at constructing an aquasystem project, which will help us future-proof Africa's freshwater ecosystems against climate change and invasive species. So before I go into the project itself, a little bit more about myself. So I was born and raised in the Eastern Cape in a small village called a Crossroads. And my first interaction with nature was us playing by the riverside when our parents were doing laundry. So that was my first introduction to the natural environment. And I guess I've been attracted to rivers since then. And I'm currently with the South African Institute for Aquatic Biodiversity, as Robert has introduced me. And that was after my PhD in botany, which I did with Rhodes University. And I am also a founder and director of a statistical consulting company called Baso R Analytics, which you have heard from Rob again about that. So moving into the actual science, I would like to start with a quote, which is from Rahel in 2008. And this says, climate change and invasive species are two of the most prevalent aspects of global and environmental change. And this is what sparked me originally from my PhD to look at both of these aspects in combination. So we know that invasive species are a problem. We've seen a lot of it here in South Africa, taking, for example, the case study at Harties, where there was a lot of invasion of water hyacinth there. Some of the attendants might know about the story. So we are very familiar with the effects of invasive species. But what happens then when you introduce another pressure or another stressor in that, which is climate change aspect? We're already struggling with polluted waters. So the addition of climate change will then proliferate or give an advantage to these invasive species even more. I'll give you one example. So if there's regular flooding, which we've seen in KZN, for example, that then means that the invasive species get time to be dispersed across to systems which they originally didn't occur in previously. So it is important to look at both of these interacting stressors in combination and not in isolation. But what I've seen in the academic space currently is that people tend to try to separate them, and I understand why, because it's very challenging to work with both of these things, and it's quite an ambitious project on my part, but I think it's doable. And you ask yourself, why invasive species? Why climate change? And as we know, in Africa, we're dependent a lot on freshwater ecosystems. I know a lot of African countries which directly use and are dependent on these ecosystems for transportation. And if you have these invasive species, then it becomes impossible to navigate those systems. And we also had Leroy, the previous doctor before me, talking about the importance of fish. And you wonder what happens when you introduce this monoculture of invasive species in a freshwater ecosystem. So that's where I come in. So I look at the trophic interactions, or what we call trophic cascades. So my question is, what happens to the aquatic life forms in these freshwater ecosystems once we remove that first layer of diversity over there? I hope you can see my where I'm pointing. So, and the primary producers, when they are simplified and you only have one producer, which is the invasive species, you would assume that in the food web, then the associated species, the primary consumers, will be simplified as well. And that will mean that the fishes then have a simplified diet. And if the species that they depend on, let's say there's a native plant species that is directly correlated with a specific fish. If that native plant is outcompeted, then you also lose that associated fish. And then that also leads to direct effects on human livelihoods, because we depend not only for water consumption, but also for fish, as I've mentioned before. So my project looks specifically at the trophic level effects. And the reason why I wanted to also contribute to this part of science is because there was a recent database which was published called ISOFresh, and I noticed there that out of all the stable isotopes, which are also called food webs in simple terms, Africa only contributed about 7% to that. And that's part of the reason why I was drawn to this work. And so going into the proposed solution a bit more. So I want to use a transdisciplinary approach, but essentially what I want to do is go out into the field and select in a participatory manner a number of sites which I can use as the baseline essentially for the model. Because the ultimate solution or the ultimate output for this project is building a model that can then be used to predict those tipping points. In sticking with the theme of the webinar today, we want to identify where in this, so is it 50% where things start to change? Is it 25% where things start to change? Where exactly are those tipping points? And we'll use a variety of invasive species, look at invaded areas versus non-invaded areas, And then in both of those instances, we want to see who is eating who in the food web. And is there simplification, as we've suggested previously, of the food web with the introduction of invasive species? And what happens when we introduce, for example, biological control? Do we see a recovery of those ecosystems and of those food webs? So we'll do that as the first layer, which is, I can see that it's not as participatory because it's mainly science. But the second part is going to be the most transdisciplinary part, is to then take those models that we've built based on those food webs to move not only from just data to enable decision making, and then move from that to impact using the Aquastar framework. And in this framework, we want to produce dashboards that will allow the users. So let's say you are a land manager and you want to know, you know your level of invasion in a specific ecosystem. So you can come to this interface here and just select the area or the region which you're working on, and also how far into the future do you want to project, going back to the effect of climate change. And also you want to, so this is obviously a very simplified version of what I'm hoping for, but you'll be able to put in here the species that you have in your system and the percentage cover of that system, and that will give you the ecosystem stability index that will tell you what the tipping points are, like how close are you to the tipping point. And also for policymakers, for example, who want like quick access to information, there will also be an option for them to just look at when you introduce a certain species and how does that then affect the trophic levels. And so obviously, as I've said, this is a very simplified version of what I'm hoping for. But essentially, when you apply that, then you'll see a change in the ecosystem stability index. And then what happens when you do some type of intervention in that. So that is the ultimate aim of the project. And I'm seeking funds specifically from the Oppenheimer generations, because I've seen how they've been able and have a track record of funding these big South African-led solutions. And I feel like this has the potential to not just solve an African problem, because we know that invasive species and climate change are a global problem. But I'd love for us to be the first ones to come to the table with solutions instead of always being the ones who are left behind, as the other panelists have already mentioned. So thank you.
Fantastic, Nompumelelo. That's really a lovely project to look at, and I'm a little bit interested in how you take some of this beyond South Africa and sub-Saharan Africa and further afield. What do you see as the prospect for engaging with other organizations elsewhere to make sure that this work can be meaningful elsewhere?
Thank you, Rob. So my aim is to just build and show that it's possible to build an infrastructure that looks at both invasive species and climate change. And I know that there's a lot of invasive species, and I can't do all of that work in this 3-year period. So my aim is to encourage people to also look at adding to the project in their own way, be it looking at specific regions of their own world, and maybe a specific invasive species of their own. Because in this current project, we're aiming to do 3 species and 3 countries within the sub-Saharan Africa. But everyone, like including Africa or any other regions of the world, are able to then build on that using their own regions, but the infrastructure would already exist, and with a clear example of how to go about it.
Fantastic. So early days on a long journey, but important work that has to be started. So thank you, Nompumelelo, and thank you to the other 2 panelists as well. We have used up quite a lot of time because we've done a couple of questions in between. I've noticed that there's quite a lot of activity in the chat. So people in the Q&A. So people have been putting questions to panelists in the Q&A, and I would encourage you to put any questions to Nompumelelo in the Q&A as well, and she can answer those over the next few minutes. We will be wrapping up at 2 o'clock, so just a few more moments left. I'd like to ask each of the panelists to just reflect again on their main motivation for this work, the real deep motivation that comes from within them to pursue this work, because it'll be that motivation that keeps one going when the going gets tough. And then also to reflect just on the beneficiaries who are most likely to be impacted by this work. So perhaps we could start. Nompumelelo will give you a chance to respond to questions in the chat first. So let's start again with Dr. Nganso. Your kind of deep why for this work, and who ultimately stands to benefit.
Thank you so much, Robert. My interest into this to develop this proposal is because when we look at the global scale, Africa is lacking behind when it comes to documentation of biodiversity data. And we do know without robust data, we cannot pass robust policy to support our food production system, which is already threatened by climate shock events in Africa, like flooding, drought event, and at the same time also conserve our biodiversity that we have in Africa. Okay, but then despite the vast land area in Africa, we are lacking behind compared to North America and Europe championing this effort. So that's where my interest coming. But at the same time, most of our local communities, even at the governmental level, they are not informed about the importance. They don't know much about the importance of insect pollinators, the landscape that support their health and their productivity. So I'm also going to unlock that by building their capacity, but also creating awareness through what I'm proposing through this project, by training, starting from the scratch, the student I'll be working with, the local guide I'll be working with in the field, the eco guard, but also the ministries on how important it is to document such data and what are the standards that should be used in documenting this interaction data, and what is the benefit of us leveraging on this to tap into new opportunities that are coming in when it comes to biodiversity conservation, like the carbon credit. When you enhance your landscape with the nutritional plants I'm going to profile, how can you leverage on that to enhance landscape suitability for pollinators? And then when that landscape is enhanced, it means you are enhancing also soil health and air health in terms of increasing carbon sequestration in the soil, and then you could benefit from carbon credit opportunities. So that is what I'm trying to address through my project. Thank you.
Thank you very much, and very powerful work. And Dr. Twoma.
Yes, so
deep motivations in 1 minute.
So if you can imagine what Africa, the challenges that we go through in Africa in terms of limited funding for research, limited infrastructure for high-tech research, limited, and with what we propose, the platform that we're proposing using the Oxford nanopore sequencer, the affordable nanopore sequencer, with what we are proposing is that trying to make Africa attain what other continents attain with very minimal cost, because we know we are limited in terms of funding, we are limited in terms of capacity in the lab. So I believe that with what I'm proposing in my project, Africa can actually contribute equally in terms of eDNA data for monitoring fish, on equal level with capable partners on the north, but with minimum cost and involving everybody, because I'm also saying that most of this thing will be co-created with all stakeholders that are involved in fish biodiversity monitoring. So imagine doing something that is being done elsewhere at a minimum cost, same standard, and involving everybody, not leaving anybody behind. I think that this will be great impact for the African continent.
Fantastic, David. Thanks very much. And then finally, last word goes to Nompumelelo. A quick one from you.
Cool. So, Rob, this is personal to me. So, looking at the way in which our freshwater ecosystems are under stress, I'm worried about the future generations. Me being a mom myself, I know the importance of protecting these freshwater ecosystems and ensuring that they last a lifetime, like not just our lifetime, but for future generations as well. So, my project will ensure that decision makers have the necessary tools to not only look at the current situation, but can look to the future as well and not plan for just the current generations, and also plan for the future generations.
Fantastic. I want to express my sincere thanks to Beatrice and Levi and Nompumelelo, all of you doing powerful and important work in your own right. And thanks for coming on to Tipping Points to share that with us. We've really enjoyed getting a little bit of a glimpse into some of the work that's being envisioned in the African continent. I'm going to move over to look forward to the next one. The next OGRC Tipping Points is scheduled for the 25th of September, and we'll be looking at some of the challenges of doing research in Africa. We're calling that Tipping Points Reclaiming Knowledge and Reshaping Impact, putting Africa first in environmental research. So please do keep an eye out on our social pages for that registration link. That will be on the 25th of September. And then also just a reminder, you can sign up for the OGRC quarterly newsletter and the JWO quarterly newsletter. And for anyone who missed part of this webinar or other webinars, please do hop along to our YouTube channel where you can watch these webinars. There will also be a feature article emerging from this webinar to share some of these insights. Once again, thank you very much to all of you for your participation. Thanks for the fantastic questions that have come through on the chat, and I'm glad that some of those managed to get answered. And we really look forward to seeing you again at another Tipping Points webinar. From now, thanks once again to our researchers. Congratulations to everyone. The winner of the JWO will be announced at the Oppenheimer Research Conference in October, on the 16th of October. So we look forward to engaging with you further there. Thanks very much, everyone. And that's a wrap from Tipping Points.